Right, hi everyone. This is Professor Francis Barr from the University of Oxford. He's going to be talking about cell division and the consequences of dysregulation. The session is recorded and the slides will be available in a few days' time, and the recording will be available on the BioTalk's BioSoc website.
Okay, so thank you for the invitation to talk to you all today. So I'm going to just tell you about one story today from my lab, just a recent one, and it's really about the way that the cell aligns and segregates the chromosomes. So I don't think we have all the answers here. Probably we end up with more questions than answers at the end of this, as you'll see. But I think it's that's what science is often about. It's about getting close to the truth, not necessarily finding answers that conform with what people expect. I can see Steve nodding. So we're interested in the process of mitosis, which is this beautifully orchestrated process by which the chromosomes, after they're replicated in S phase, are captured by the microtubules of the mitotic spindle and then segregated out to the two daughter cells. So this is a really prevalent process in our bodies. New cells are created through the cell division process, and it operates with a very high degree of fidelity. And there are lots of complex details about this which I don't want to go into today. Just to say that mitotic spindles are precisely sized with respect to the number of chromosomes that have to be segregated, and these alignment processes is tightly coordinated with segregation of chromosomes. So chromosomes have to align before they can segregate, and then cytokinesis can only really be triggered once the chromosomes have segregated. So that process is controlled by a whole host of factors, and the ones that we're most interested in are protein kinases. So there are three protein kinases that are kind of relevant for my talk today, and they're kind of examples in a way of the different types of protein kinase activities within the cell. So we have the Aurora A protein kinase, which essentially rides on microtubules, and this is thought to regulate microtubule dynamics and the formation of the mitotic spindle. We have a related kinase, Aurora B, and Aurora B localizes to chromosomes themselves because it's got a DNA binding module, and it regulates the structure of chromosomes and the process of cytokinesis. And this third protein kinase is a kind of a signaling molecule which regulates whether cell cycle progression can occur, and this binds to kinetochores conditional on their status. So the general idea is that Aurora A promotes the formation of spindles and mitotic progression. So if you inhibit or interfere with Aurora A activity, and there's a whole host of genetics and cell biological studies that show this, you get small mitotic spindles, and the cell arrests because it can't finish this process of chromosome alignment properly. If you look at the role of Aurora B and this kinase MPS1, they seem to work together, and they're important for a pathway, the so-called spindle assembly checkpoint pathway. So while chromosomes are aligning, this checkpoint is active, and the cell is prevented from going into anaphase and segregating its chromosomes. And if you inhibit Aurora B or MPS1, that checkpoint mechanism fails. So you kind of have a simple idea where Aurora A promotes spindle formation, Aurora B and MPS1 check whether that process is complete. Once it's complete, their function is ended, and then the cell goes into anaphase and segregates the chromosomes. So just to kind of go through this in order. So if you have a cell in G2, it has a centrosome, which is the microtubule nucleation point for the spindle poles, and you have the decondensed chromosomes in the nucleus. Kind of first step of mitosis is really before the nuclear envelope breaks down, you have separation of the centrosomes, the spindle poles start to form and move apart, and this is where Aurora A function kind of starts. Then you have chromosome condensation, which I haven't shown you, but you get condensed chromosomes. And this condensation process does depend on Aurora B, but the key spindle formation process itself is driven by Aurora A. Now, these chromosomes have Aurora B and MPS I localized to them. So Aurora B localizes to the centromere region, so cartooned it in green, and the little red dots indicate that MPS I is also recruited and sending out this checkpoint signal, which is preventing exit from mitosis. Then the chromosome alignment process, this can only really occur once the spindle has formed, so the chromosomes become captured, and this is to some extent regulated by both Aurora A and B. But as long as you have a single chromosome which isn't attached, the checkpoint signal mediated through this protein MPS I is on, and the cell can't leave mitosis. So only once the last chromosome is captured and the checkpoint is off can the cell think about leaving mitosis, which again leads to further movement of the chromosomes, so they segregate to the cell poles. And this, to some extent, the structure of the spindle depends on both Aurora A and B. But the role of Aurora A is probably related to chromosome segregation, and the role of Aurora B is rather about the process of cytokinesis, so cell division. And if this goes wrong, which I don't want to labour the point, but if you get missegregated chromosomes, and there are lots of different ways that chromosomes can be missegregated, they tend to get partitioned off into small structures, which we generally refer to as micronuclei. Although I would say some micronuclei are not really single chromosomes, they're much larger structures. So in the literature, there's sometimes confusion between fragmented nuclei and micronuclei, which we should probably be a bit more careful about. So if you think of the kind of some more of the mechanistic details, what's happening. So initially, chromosomes are condensed and they're not captured by the mitotic spindle, and they're checkpoint signaling active. And that's because they have Aurora B on the centromere and MPS I localized to the so-called kinetochore. So the kinetochore is the microtubule attachment site, and I don't want to go into the mechanism, but just to say when microtubules bind to kinetochores, MPS I leaves. And this obviously, in order to align the chromosomes on the spindle, both of the kinetochores have to bind a microtubule. So you need to establish this kind of geometry where a chromosome is attached to both spindle poles through its kinetochores with this kind of opposing geometry. So to achieve this, the cell typically makes and breaks a series of microtubule attachments to the kinetochores, and that process, that error correction cycle, is in some way driven by Aurora B and MPS I. And in this model, the idea is that the checkpoint active kinetochores essentially have a high Aurora B phosphorylation state, and as they reach correct attachment geometry, the phosphorylation diminishes, and then at some point a stable attachment geometry is reached, which is further stabilized by recruitment of other factors, which are the asterin complex and the scar complex. And this final geometry can resist high tension and essentially doesn't tend to re-enter this cycle. So in this model, Aurora A doesn't really play a direct role in these processes. However, it is important for the formation and the stability of the microtubules which attach to the kinetochore. And Aurora A is really, it's not really a single complex, so it's a series of activities together with different partners on the mitotic spindle. So I'm going to focus on this one today, but that's not to ignore the others. So the thing about this cycle, and probably about all protein kinase function in the cell, is if you have a kinase, you almost invariably need a phosphatase, because as I've diagrammed here, you tend to have cycles of regulation. So if you're adding phosphorylation, you need to be able to remove phosphorylation. So protein kinases are really crucial regulators, but they always tend to work with protein phosphatases, which are equally crucial regulators. So we tend to think of protein phosphorylation, dephosphorylation cycles in terms of substrates. So you have a substrate which needs to be regulated and a protein kinase which is active. So for example, Aurora A, and it phosphorylates that substrate using ATP, and then that phosphorylated substrate can be dephosphorylated by a protein phosphatase, so a PPP. Now, you know, the sharp-eyed amongst you will have realized that protein kinases, as I've diagrammed here, are often also phosphoproteins. Okay, so protein phosphorylation is typically associated with protein kinase activation. So Aurora A is made in an inactive form and is activated by phosphorylation, an autophosphorylation reaction, when it binds to one of its binding partners. So for example, TPX2. Now that presents a further opportunity for regulation by protein phosphatases. So protein kinases themselves are substrates for regulation by protein phosphatases. So in a protein kinase phosphatase system, you typically have the kinase, which is being regulated by a phosphatase, and then the substrates of the kinase are regulated by that kinase and counteracting phosphatases. So there are multiple roles for protein phosphatases in these systems. So if you thinking of process of mitosis, there are many protein phosphatases in the cell, but thankfully most of them don't seem to play a role in cell division. So the major family that we're interested in terms of cell division is the so-called PPP family, and they have very similar catalytic subunits that can be broken down into two broad classes. So there's the PP1 family, which has a highly conserved series of 3 catalytic subunits that form interactions with single so-called regulatory subunits or dock directly to their substrates. And then you have these multimeric enzymes, which typically have 3 subunits that come together to recognize specific substrates. So these enzymes here all share the same catalytic subunit, but they just have different regulatory subunits. PP4 and PP6 have different catalytic and different regulatory subunits. And these enzymes here share a common so-called scaffold subunit, which is often mutated in tumors. And this somehow alters the balance of these enzymes in tumor cells. But that's really very poorly understood. So the phosphatase that I'm going to focus on today, and I think it provides a kind of example of phosphatase regulation in cells, is especially in mitosis, is this enzyme PP6. So PP6 we showed together with Ulrike Gruneberg's lab many years ago now, is mutated in malignant melanoma and is the counteracting phosphatase regulating Aurora A. So this enzyme is a trimer. We've mapped all the subunits you need for this. I don't want to explain that today, but this is a distinct enzyme which regulates the phosphorylation level of Aurora A. So Aurora A is activated by binding to one of its activating partners. The one that we've characterized most is TPX2, and this active enzyme can be dephosphorylated by PP6. And PP6 is interesting because the catalytic subunit's mutated in melanoma, and this is thought to act as a driver for Aurora A activity, which amplifies its activity in melanoma cells. And we've shown this, and other work in the literature supports that view. So if you look at, or we did a number of years ago, you survey mutations through the PP6 gene. This is just a subset of those mutations, and you can see all of them reduce the phosphatase activity, either partially or nearly completely. And the Aurora is a phosphoprotein. It runs as a doublet, and you can see in cells which which carry a loss of function mutation in PP6, the lower unphosphorylated form of the protein is lost, and you only have the upper phosphorylated form of the protein. So you completely shift the equilibrium towards only active or mainly active Aurora A. And these cells, when you look at them, this is a melanoma which has a wild type PP6 function, has mutations in the RAS-MAP kinase pathway. This is a melanoma which has mutations in PP6. And you can see here, these cells are segregating their chromosomes normally, but they're tumor cells. These are tumor cells which don't segregate their chromosomes normally, and they form these abnormal nuclei with some micronuclei. And as many other people have observed, these micronuclei, you can't say in all cases, but in many cases are associated with DNA damage. So the idea has arisen that this kind of genetic change generates a genome instability phenotype through amplification of protein kinase activity, and this drives tumor evolution through a kind of chromosome damage cycle. So some people refer to this as chromothripsis. Whether this is chromothripsis, I don't think we can say. So chromothripsis is a kind of chromosome break and rejoining cycle, but we think that this probably is a kind of example of chromothripsis. Now, the thing about all this work, and for a long time I think we struggled to make progress here, was although we knew these things are happening, the molecular targets of Aurora A, which explain this phenotype, were not clear. So as I've said, they could have been proteins on this mitotic spindle, but of course the protein kinases can have many substrates. So a phenotype like this, which is actually quite complex, you could take a candidate approach and screen substrates, but that's got some problems. It's a bit biased. So we basically then set out to look at that in more detail. And before I show you that data, I just want to tell you one thing which you need to remember when I'm telling you everything else I remember. So this is chromosome segregation in normal cells at the top. You see neatly lined chromosomes with a nice defined spindle, which segregates chromosomes beautifully in tightly defined plates in anaphase. If you look at cells which are knocked down for PP6, you can see the spindle appears much more disordered, and the chromosomes don't align and compact so nicely into the metaphase plate, and the chromosome segregation in anaphase appears much more disordered. And you can see here some of the chromosomes are actually escaping the spindle, and we actually track these in this cell. They go off and they go into a large micronucleus or a nuclear fragment, depends on your definition. So this is kind of just a summary of what I've said. So we have a system where Aurora A activity is amplified in tumor cells because of loss of PP6 through mutation, or we can engineer in the lab. This kind of recapitulates the phenotype seen in other tumors where you simply have Aurora A amplification. So either Aurora A amplification or loss of PP6 give the same outcome, which is genome instability and acts as a tumor driver. And we think that these cells, and we have some evidence for this, can only survive because of the spindle checkpoint pathway. So, the substrates of Aurora A became kind of really crucial for us to understand. So a few years ago, I persuaded two of my master's students who were doing their degrees in biochemistry to embark on a slightly different approach, which started off with this. So we made some new cell lines using CRISPR, and what we found was that when we knock out PP6 and we characterize the cell lines, all of the cell lines have nuclear morphology defects. So these cells are all kind of, they all have problems segregating their chromosomes. and they have problems generating normal nuclei. And they're all knocked out for PP6. And when we arrest them in mitosis, we can see that compared to the parental cells, they all have elevated levels of Aurora phosphorylation. So this is the parental cell line, and on the short exposure, you can't actually even see Aurora phosphorylation. But if you knock out PP6, you see elevated levels of Aurora phosphorylation. If you then arrest the cells in mitosis, you start to see the Aurora phosphorylation in the parental cell, and this is further increased in the knockout cell lines. So this was a good start. We could make nice CRISPR knockout cells. These are all genomically unstable. So as you grow them, they become more and more different, which is why you need multiple cell lines, because you want to be sure that you're getting the same result in different backgrounds, not simply in one cell clone. But all of these cell clones, when you characterize them, have the same change. They have abnormal nuclear morphology, and they have hugely enlarged mitotic spindles. Now, they still have the, and this increase in spindle size depends on Aurora A activity. So if you take these cells with their large spindles and you treat them with Aurora A inhibitor, the spindle gets smaller again. So this looks like an Aurora A amplification phenotype. And you can see here the spindle still has two poles, okay? So you haven't lost spindle bipolarity. And there's lots of other literature showing that Aurora activity, A activity, is in some way important for regulation of spindle size. Obviously, it's important there. We're talking about a genome instability phenotype. So the first thing you've got to check is, do the cells actually have the same number of chromosomes? Because spindles get bigger if the cell has more chromosomes. So if you have genome instability, you have more chromosomes, you might get bigger spindles. So actually what we see is parental cells, these are actually parental HeLa cells. They have some slightly ill-defined number of chromosomes, but it's fairly fixed. And PP6 knockout cells, they actually have reasonably nice chromosomes, but they have some chromosome fragments. And you can see on a FACS plot, these cells actually don't have more chromosomes. They're actually drifting rapidly to smaller chromosome number. And of course, that fits with the idea that they're damaging their chromosomes. They're damaging the genetic material. They're actually losing genetic information. They're not gaining more information. And we used a slightly different assay here as well to test for spindle size, where we give a drug that turns the bipolar spindle into a monopolar spindle, and we still see an increase in spindle size in the PP6 knockout cells that's sensitive to Aurora A inhibitors. So this tells us that there's some kind of fundamental change in the ability of the cell to build a spindle of the correct size, and that doesn't appear to be a simple spindle bipolarity phenotype. The spindles are bipolar, and even if we collapse them into monopoles, we still see a size difference. So that to us suggested that some process relating to either microtubule organization or the interaction of microtubules with the chromosomes was altered. And we could observe that there were some molecular changes. So this is a protein aspirin, which is normally, as I've said, it's part of a complex that associates with stable microtubule attachments to kinetochores. So you see here, this is a bipolar spindle, and it remains attached to the kinetochores in anaphase as the chromosomes segregate. And if you get rid of this protein, the spindles become much more disorganized, and chromosome segregation in anaphase is perturbed. And what we saw in the PP6 knockout cells was this protein doesn't really accumulate properly on kinetochores. It's kind of just hanging around on the spindle poles, and it stays there in anaphase. And if you inhibit Aurora A, you can kind of partially rescue that phenotype, although obviously the spindle is kind of tiny because giving Aurora A inhibitors shrinks spindles. So this told us that there was something wrong in the spindle formation process, but doesn't really tell us. And it tells us it's Aurora sensitive, but it doesn't really tell us what it is. And these are just some example live cell imagings of these knockout cells. So compared to matched isogenic parentals. So parental cells align the chromosomes and then segregate them normally. And the knockout cells align the chromosomes, but badly, and then segregate them in a very diffuse kind of way. And then the chromosomes often escape from the kind of anaphase spindle and form these kind of large nuclear fragments, or sometimes micronuclei. And the cells also take much longer to go through mitosis. So we wanted to understand what are the Aurora A targets that really explain these changes. And to do that, the students in the lab, so Stefanos initially, then Samvid, working with Samvid, and then a bit later on Colette. I mean, I say this, this is actually because none of my postdocs would work on this. I think they all thought I was nuts, which is, I think, a lesson to everybody. The students were willing to take the risk and do some exciting new science, and my postdocs were a bit too play it safe. Anyway, so our idea was that parental cells build a spindle which kind of compacts the chromosomes, which then segregate in anaphase, and you get nice normal nuclei. PP6 knockout cells are defective in this, chromosomes escape, and you get micronuclei. And that has some consequence for their growth. So we should be able to, by doing a genetic screen, find suppressors that essentially correct the chromosome missegregation defects and maybe also suppress the growth defects. And we also should be able to find synthetic lethals which kill these cells preferentially, either because they make mitosis less efficient or because they identify a pathway in micronuclei which kills, can kill the cell. So this is just a quick summary of the screen. So we engineered EHAP. So this is a haploid genomic screen to remove the PP6 allele. That knockout creates cells which are micronucleated. They lack PP6. This is just a Western blot, and they have elevated Aurora A activity, as you'd expect. We then infected them with a lentiviral genome library, so 120,000 guides, 5,000 copies of a guide per gene, or six individual guides per gene, or 5 to 6. So then you essentially kill all the uninfected cells with puromycin, leaving only the CRISPRed cells or cell population, and then you do essentially a growth assay. And at the end of that growth assay, you recover the barcoded CRISPR guides using PCR, you sequence them, and then you put your data set back together. So what you can do here is you can ask, is there positive or negative selection for individual genes in a wild type or a PP6 knockout background? So this is just a plot which says if you take all of the data, it doesn't make any difference if you have a PP6 wild type or PP6 knockout cell line, and you do this. And that makes sense. So there's no gross genetic change in a PP6 wild type versus a PP6 knockout cell if you take all of the genes. And these are just 2 replicated screens. The reason this peak here, which is centered roughly around 0 effect size, that's all genes. And this slightly offset peak here, these are the so-called core essential genes, which show a stronger level of selection. And minus essentially tells you they're essential genes. So this told us our screen worked. So then what you do is you take the screen data from the wild type screen and the PP6 knockout screen, and you ask which genes show a differential effect. Okay, so that gives you something like a volcano plot, So on the left, we have genes which are differentially selected negatively, so candidate synthetic lethals, and these are so-called candidate suppressors on the right. And most things in this type of plot, because of the nature of the screen, cluster around kind of the origin, because most genes show the same effect size in both the parental and the knockout cell line. Okay, so we're not expecting most things to show the difference, and they don't. So we identified all of the subunits of a single chromatin regulating complex, which I'm not going to talk about because we're still working on that. And we found the kinetical protein NDC80 as a reproducible hit in this screen. And then on the other side as a suppressor, we found the Aurora A regulator TPX2. So that to us made some kind of sense. So first of all, I just want to talk about TPX2. So TPX2 is the activator of a pool of Aurora A that sits on the mitotic spindle. So if you lose PP6 function, you have more Aurora A. So if you reduce the amount of TPX2, which is the thing that's activating Aurora A, you should essentially suppress the phenotype. And that's what our genomics data kind of tells you. So I don't want to talk through all of this slide, but just focus here and on particular on this graph. So if you take a cell and you introduce if you ask, does TPX2 play a role in controlling spindle size? The answer is yes. So these are cells where we've removed TPX2 and the spindles get smaller. If we add back a wild type TPX2, we can recover the spindles to their normal size. And if we add back a mutant TPX2, which can't bind Aurora A, the spindles are small again. So TPX2 plays a role in spindle size control, and that role requires its ability to bind Aurora A. And there are some Western blot controls here that I won't discuss further. The key experiment that we wanted to do was then to ask, okay, so does this help explain what happens in our genetic screen and in the PP6 mutant or knockout cells? So here we're looking at spindle size in parental cells and PP6 knockout cells, and we're exploring the role of TPX2. So if you look at spindle size in a parental cell or a PP6 knockout cell, you can see the spindles are larger when we knock out PP6. If you then knock down TPX2, they tend to smaller size and roughly the same small size. And that's true whether we look at bipolar spindles or we look in the monoastral spindle assay. And this role of TPX2, this is a bit of a complicated slide, so this is just the microscopy data. And this is looking at Aurora phosphorylation. The key thing to focus on is this graph here, which is looking at spindle size. So in a control cell, we have a spindle size here of about 10 micrometers. Under these conditions, in a PP6 knockout cell, that's increased significantly. If we knock out TPX2, the spindle size goes to a lower value in both cases. If you put back wild-type TPX2, you can recover the spindle size to the control value in both cases. So smaller size in a parental cell or bigger size in a PP6 knockout cell. And that increase depends on the ability of TPX2 to bind Aurora A. So this mutant can't bind Aurora A. So we kind of come to the conclusion that the suppression of the phenotype that we're seeing in the assay is because TPX2 is driving Aurora A activity in the absence of PP6, which is, sorry, it's high tech. That's why TPX acts as a suppressor. Now, you might say, well, that the selection here isn't particularly strong, and I think that's a fair comment. But the reason is we're not saying these cells are normal. What we've done is we've taken away PP6, which is a regulator of Aurora, and now which is reducing Aurora, which is amplifying Aurora activity, and now we've taken away the thing that was helping to localize and activate Aurora. So these cells, although we've partially suppressed the phenotype, they're by no means normal cells, but it does, in this kind of assay, rescue part of the growth defect. So we're not saying they're normal cells, we're just saying that in genetic terms, these cells are now more fit than they were. So what about the other side? What about NDC80? So this is a much stronger phenotype. As you can see, it's shifted further away from the origin point than this weak suppression phenotype. And you can see that if or you can understand that very clearly if I show you these images. So these are cells which are parental, so wild type PP6 or knocked out for PP6. And you can immediately see the kind of crude phenotype. Nuclei are quite malformed due to mitotic defects. If we knock down NDC80 in these cells, so this is not a knockout, it's just reducing the concentration, you can see the cells start to have chromosome segregation defects and form micronuclei after about 48 hours. If you do that same 48-hour knockdown in a PP6 knockout cell, you see these really drastic changes. So these cells are really unable to carry out mitosis normally at all. So PP6 knockout cells are very, very sensitive to the levels of NDC80. So what's NDC80? So NDC80 is the major microtubule binding protein of the outer kinetical. It's actually part of a heterodimeric complex with a protein called NUF2, which I'm going to tell you and then ignore for the rest of the talk. So as far as we're concerned, NDC80 is a long extended protein. One end binds to the chromosome and the other end sticks out into the cytoplasm and binds microtubules. And this binding, according to the literature, is regulated by Aurora kinase phosphorylation. And in particular, it's been mainly attributed to Aurora B. And it's also phosphorylated by CDK cyclin B, although the role of that's not so clear. And for those of you who are interested, this molecule exists as a kind of cluster on the surface of the chromosome. So if a microtubule comes towards the chromosome, so that's looking at the end of a microtubule, so you can see a ring of tubulin heterodimers, and these blue things, they're the heads of the NDC80 molecule kind of clasping around the end of a microtubule. So this is really thought to be the kind of fundamental complex which explains how a chromosome latches on to a microtubule. And the current literature has kind of, and actually there's a textbook model, so this is from molecular biology of the cell, or a variant of model in molecular biology of the cell. So the idea is that if a kinetochore doesn't have microtubules bound to it, it's heavily phosphorylated by Aurora B, and the Aurora B phosphorylation is thought to be on the N-terminus of this NDC80 molecule close to the microtubule binding site. And as the microtubule binds, this whole assembly is pulled away from the surface of the chromosome, so this sort of brown thing here, and it escapes the Aurora B complex, which is actually localized on the chromatin. So the kinetochore goes from a high phosphorylation state to a dephosphorylated low phosphorylation state as microtubule attachments form. And this is thought to be important for the stabilization of microtubule binding. So here, microtubule binding is actually low affinity because of the phosphorylation, and the affinity increases because of the application of force, in part because of the removal of phosphate. And this has kind of informed a kind of model in the literature, which was, I think, initially proposed by Lampson and Kapoor, and has been then kind of elaborated by others, which says that normally this cycle is driven by Aurora B. So if microtubules attach, you get this force generation and you get dephosphorylation of the kinetochore. But if a chromosome were to drift close to a pole, it would meet Aurora A, and Aurora A would phosphorylate the kinetochore in a tension-independent fashion, release the chromosome from the microtubule, and it would get pushed back into the middle of the cell. So we thought, well, the reason we're getting NDC80 in our screen is because, in our case, we've removed PP6, and now instead of having a gradient of Aurora A, where it's quite tightly packed around the pole, it's just distributed through the whole spindle. And chromosomes essentially go on a mad dance, and they can't work out where they are because there's no phosphorylation gradient. So this seemed to be a really good idea. And of course, you already know that I'm going to tell you it's not true. But, you know, it seemed to explain absolutely everything that we'd seen. So we then wanted to ask these questions, or answer these questions. So we wanted to know, is NDC80 phosphorylation actually increased in PP6 knockout cells? You know, what are the contributions of Aurora A and B to this phosphorylation during spindle formation? Does the phosphorylation actually play any role in spindle size control? And does deregulation of this phosphorylation explain the phenotype of these knockout or cancer mutant cell lines? So the first question, kind of easy to answer. So this is just some Western blot analysis of parental and PP6 knockout cell lines. So this is a phosphor antibody to a conserved Aurora phosphorylation site in NDC80, and you can see in mitosis, the level of phosphorylation is increased if you knock out PP6, goes up about threefold in relative terms. This is just a different way of looking at that using a method called phostag, where the phosphorylated protein is retarded as it moves through a gel. So you can see there's some phosphorylated species in this parental cell line, and it's shifted up. So there's essentially more phosphorylation on the protein if we knock out PP6. And that increase is, obviously we would predict Aurora A dependent. And just for the skeptics, or to share good practice with you, I think it's always really important to do lambda phosphatase controls for phospho-specific antibodies. So if you take the same sample, you treat it with lambda phosphatase, does your phosphor antibody signal go away? So here you can see it does. So we know that this signal and this increase is really phosphorylation specific. Now, as I introduced at the beginning, phosphatases could play two roles in this system. So one role could have been that actually PP6 was the phosphatase for NDC80. And the other explanation would be that PP6 is actually just the phosphatase for Aurora A. So we then, using a phosphatase assay, we checked Aurora dephosphorylation in PP6 knockout cells. So parental cells, it's dephosphorylated, knockout cells, it's not dephosphorylated. And we looked at NDC80 dephosphorylation. So these are just normalized to be on the same scale because the knockouts should actually be at 3, not 1, but we just normalized to 1. But you can see dephosphorylation of NDC80 has different kinetics to Aurora A and is actually not PP6 sensitive. So the contribution of PP6 here is because it's creating a situation where there's more Aurora activity, not less NDC80 dephosphorylation. So is that increase actually explained by Aurora A activity? And given the background to this is we would expect that the basal level of phosphorylation is probably being carried out by Aurora B, and Aurora A is just increasing it. So we then looked at Aurora phosphorylation of NDC80 in cells, and this is just some Western blot data using specific inhibitors. So if we add an Aurora A inhibitor, we essentially lose most of the NDC80 phosphorylation. If we add Aurora B inhibitors, it has little effect on NDC80 phosphorylation. And if you mix them together, you get just the same result as having only Aurora A dephosphorylation. And you can see here the Aurora A inhibitor is inhibiting Aurora A because you lose the activating mark on Aurora A. And if you look in the cells, is the phosphorylation in the correct place? So this is just the pattern of kinetochores here, and this is the pattern of phosphorylation. If you add, and you can see this is on kinetochores as the spindle forms. If you add Aurora A inhibitors, it's gone. If you add Aurora B inhibitors, it's still there. And if you add both the inhibitors together, it's still gone. And if you look in a PP6 knockout cell, you get largely the same pattern. It's just the signal is slightly stronger. You can do this with mass spectrometry. So we mapped all the phosphorylation sites on NDC80 using mass spectrometry, and there's a kind of cluster in the N-terminus. One of the sites at threonine 31 is actually a CDK site, but the rest of these sites are Aurora consensus sites and appear to be Aurora sensitive. So this is just a Western blot experiment of the same sample, so we can see loss of serine 55 as a diagnostic mark, and mass spectrometry gives the same result. Threonine 31 isn't changed by Aurora inhibition because it's not an Aurora site. And the key thing I want to stress here is, and I hope you've been looking at the slides carefully, is it doesn't matter if we inhibit Aurora B. There's no change in NDC80 phosphorylation. It's only when we inhibit Aurora A. So you have to remember this is a phosphorylation that was presumed to be, or phosphorylation of this protein was presumed to be driven by Aurora B. And I probably should skip over this, but I just want to spend a little bit of time on this. We then checked other phosphorylation sites using antibodies, and they showed the same behavior as serine 55. But I just want to stress that the Aurora A and B inhibition is absolutely specific under the conditions we've chosen here. So if you add an Aurora A inhibitor, you lose Aurora A activation mark, which is threonine 288 phosphorylation. If you add the Aurora B inhibitor, you don't change it. This is a pan antibody which sees Aurora A, B, and C, the activating mark on them. And you can see here, Aurora A inhibition, you lose the Aurora A activation mark, but you don't affect the Aurora B activation mark. If you add an Aurora B inhibitor, these are two different inhibitors, you lose the activating mark on Aurora B, but you don't affect the activating mark on Aurora A. And if you add them together, you lose all the activating marks. So clearly here we're dissecting out Aurora A and B activity, and we're showing that only Aurora A activity is required for NDC80 phosphorylation. And this is just a kind of functional test of Aurora B activity in cells. So as I told you, the Spindle checkpoint requires Aurora B activity, and we can test for that in cells. So if we add an Aurora B inhibitor, the same conditions as I've shown you, you can see rapid loss of Aurora B activity in the cell. And you can see here, there's also rapid loss of checkpoint markers from the kinetochores compared to the controls. And if you add Aurora A inhibitor, that doesn't affect the checkpoint. If anything, inhibiting Aurora A makes the checkpoint more active. So we're sure that we have functional inhibition of Aurora B in the cell. And you can do the same experiment in a different way. So you can do sRNA depletion of Aurora A or Aurora B, and you can see the Aurora A depleted cells lose kinetochore phosphorylation of NDC80, but they don't lose NDC80. And if you deplete Aurora B, you keep NDC80 on the kinetochores, and it's still phosphorylated. And nicely, you can see here, we've depleted Aurora A, and you can see the spindle size is much smaller because all the kinetochores are clustered. And actually, if you deplete Aurora B, if anything, the spindles get kind of the chromosomes spread out for some reason. And you can do the same thing using a kind of biochemical or biochemical/cell biological approach, which is if we remove the activator TPX2, we reduce the pool of Aurora A on the spindle, and so we lose TPX2 from the cell, but we also greatly attenuate phosphorylation of NDC80 at all of these residues in the N-terminus of the protein. And the kind of final, I think, nail in the coffin for the existing model, the textbook model, is this data here, where if we look at the pattern of kinetical phosphorylation, we see that kineticals, some of the kineticals are phosphorylated in prometaphase as the spindle forms. It looks like all of the kineticals are phosphorylated in metaphase, and they remain phosphorylated in anaphase. And that impression is substantiated by the Western blot experiment, where we see phosphorylation during spindle formation that persists into anaphase and then is only lost in telophase. And that's a phospho blot, and that's a phostag gel. So what you have to think about here is the prevailing model has been phosphorylation should be present in prometaphase. It should be lost in metaphase and completely gone in anaphase, because it's associated with checkpoint signaling and error correction. So just to look at that more carefully, we then asked what is the relationship between microtubule attachment and the phosphorylation reaction itself. So what we did was we used this marker for attached microtubule attached tensioned chromosomes, astrin, and you can see here these are the attached kineticals, and there's a good correlation between phosphorylation of the kinetochore and microtubule attachment status. And this fits nicely with the localization of Aurora A. So this is Aurora A in the spindle, and these Aurora A positive microtubules or spindle fibers actually contact the kinetochore. So rather than phosphorylation occurring by Aurora B at microtubule-free kinetochores, what we think is happening is that we have phosphorylation upon microtubule attachment by Aurora A. So the microtubule attaches to the kinetochore, delivers Aurora A, and it phosphorylates the NDC80 protein. And if you look at Aurora B, and you can see this is actually an anaphase cell here. So the Aurora A remains on the spindle and closely associated or proximal to the kinetochores, even in anaphase. And this white band here, that's Aurora B. And even in metaphase cells, Aurora B is on the centromere, which is clearly resolved from the kinetochore. It sits close to the kinetochore, but is actually not, it doesn't really overlap with the kinetochore. So it's close, and I think guilt by association, you could say it's close enough to phosphorylate it, and I would agree with that statement. However, as far as we can tell, Aurora B does not do the phosphorylation. It's carried out by Aurora A. And if you look at checkpoint signaling, what you can see is that this is the simplest experiment to understand. You can see this is checkpoint active kinetochores in red, and we have phosphorylated kinetochores in green. So the green ones are microtubule attached, and the red ones are microtubule free. And you can see the red and the green don't overlap at all. So checkpoint signaling and microtubule attach and phosphorylation are mutually exclusive, and that's what you would expect for an Aurora A-dependent process. So in prophase, the kinetochores are not phosphorylated, and they start to attach to the microtubules and align in prometaphase. So here they're all checkpoint active and preventing entry into anaphase. As they attach, they become phosphorylated and the checkpoint silences. Then they're fully attached, or they're all phosphorylated, and the phosphorylation is maintained into anaphase. I should probably just skip a couple of slides here and just kind of move to the end. So what we then wanted to understand was if we took this what we'd learned, really was NDC80 the key functional target that explains why PP6 mutant cells with amplified Aurora activity have large spindles and missegregate their chromosomes. So to do this, what we did was we made a series of cell lines which are matched, and they either have gene replacements for NDC80 as a wild type protein with Aurora phosphorylation defective 9A mutants or phosphomimetic mutants. And you can see here that 9A mutants actually have smaller spindles. 9D mutants actually have bigger spindles, just like the effects of amplifying Aurora A by deleting PP6 or inhibiting Aurora A with a drug, so the spindles get smaller or bigger. And then if we use these cells for a functional test, we can show that if we deplete PP6 in the wild type condition, you get abnormal nuclear morphology. If you deplete PP6 in these 9D cells, you make the chromosome, nuclear morphology and chromosome segregation defects much worse. And if you use the 9A cell line, these defects are suppressed. So the cells are not completely normal, but actually it looks from these experiments that NDC80 phosphorylation by Aurora A really is a crucial weakness which explains the chromosome missegregation and spindle size defects. So we kind of moved to a different view to the prevailing view. So what we think is that Aurora B carries out phosphorylations on the centromere and kinetochore, which are crucial to drive the checkpoint, but those phosphorylations are not in the microtubule binding domain of NDC80. When the microtubule attaches to the kinetochore and force is generated, this attachment process leads to Aurora A delivery along microtubules, and that phosphorylates the NDC80. And that phosphorylation seems to be crucial for the maturation process, which leads to recruitment of proteins like asterin and the SCAR complex. So phosphorylation appears to mark microtubule attached kinetochores and help promote a kind of affinity maturation, recruiting other proteins. So if you go back to this kind of model, we started off with phosphorylation going down, and what we now think is phosphorylation is actually going up. So obviously there are many things we still don't understand. So we don't really understand yet how phosphorylation regulates the recruitment of these proteins. But what we do know is that the NDC80 is the key target, which explains how that works. So we need to understand the mechanism. And we still don't know really which protein Aurora B phosphorylates to drive recruitment of the checkpoint kinase MPS1. So as I said, this work was started by Stefanos, Samvid and Colette, who were undergraduate project students or master students, and finished by a great postdoc, Tomoaki, with help from Cassia, Luke, and another PhD student, Kalo. And we started this in collaboration with Ulrike's lab in Oxford. So thank you. And thanks to our funders.